Hello and welcome to Science Research Weekly, Season 4, Episode 1, Year of the Mollusk. So it's a new year, so I'm trying some changes to the format. Expect more pep, video work, and hopefully better audio experience. Also trying to grow my podcast to do all that audience engagement thing like sharing with others, following the channel, and the like. So for this episode, get ready for RNA lanterns, fungal batteries, punk rock mollusks, supercharged hornworts, the top 10 do loop posts of 2024, and a Trailblazer award from NIH. This episode covers materials from January 3rd to January 9th. Let's get started with some research articles. First up here is a modular platform for bioluminescent RNA tracking. So, researchers were able to create RNA tags that recruit light-emitting luciferase fragments, which they gave an awesome name, RNA lanterns, which allowed for tracking RNA in both cells and live animals. The tagging was also sensitive enough to be detected with only a single copy. Next up, 3D printed cellulose-based fungal battery. Researchers combined two species of fungus with cellulose nanocrystals, nanofibrils, adding in carbon black and graphite flakes, yada yada, to essentially make this electrically conductive printed ink. So they could print it out. I think there's a great picture here. So they could get this all together, print it out in the ink, and make these microbial fuel cells, or you could also call them biobatteries. So four of the batteries, at least they claim, could power a small sensor for several days, so 65 hours. Kind of interesting stuff there. All right, moving right along here, we have Redox-activatable, inhalable, mucoadhesive proteinic nanotherapeutics for targeted treatment of lung cancer. Wow, there's a lot of words in there that I've never heard before, especially not all in the same context. Anyways, researchers used adhesive proteins from mussels, so the type of mollusk, not the cell type. So they use this mussel protein to improve cellular uptake of inhalation-based lung cancer treatments. The idea is so that patients can actually inhale the treatment directly. I think there's maybe a picture here. Yeah, inhale directly, like with almost like an asthma type inhalation. But then the treatment will actually stick and stay on the lung cells using this muscle protein. They won't go back out. So very interesting thing there. Muscles are really doing a lot of work here. Speaking of muscles, we have next paper, new Silurian aculiferan fossils reveal complex early history of Mollusca. So mollusks, they're really complicated. They're actually the second most species-rich animal phylum. And I'm learning how complex they are because I'm dabbling with some higher level taxonomy, and it's very messy. And mollusks are very old. They stretch back into deep time where haze obscures their evolution. Researchers have recently found two really well-preserved species from the Silurian from this, the Herefordshire Lagerstätte, so that's probably in England. And the two species have really well preserved. They have 3D imaging and everything. They've nicknamed them actually Punk and Emo. Actually, scratch that. Their bona fide scientific names are Punk ferox and Emo vorticacadium. So that's literally their name, Punk and Emo. These both fossils are actually really strange and come out from one of the clades. So the aculiferan that were really actually thought to be much more conservative in body form. So this really throws a wrench on that idea. So it turns out mollusks are lots more weirder and stranger in the various clades in the past. All right, we'll move along to electrical signaling and coordinated behavior in the closest relative of animals. What is that relative? Well, it's the tranoflagellate Salpingosia rosetta. So these are single-celled eukaryotes. Tranoflagellates are very closely related to animals, so metazoa. Anyways, researchers found evidence of actually electrical signaling to spread information between these eukaryotes. So similar to how multicellular animals coordinate their multi-cells. So kind of interesting there of like maybe where that came from or such and such. All right, we're moving along here with, there's a lot of really great articles this week, so that's why we're getting through so many articles today. Anyways, there's repetitive, there we go, injury induces phenotypes associated with Alzheimer's disease by reactivating HSV1 in a human brain tissue model. So they researchers used a 3D human brain tissue model. They mimicked a concussive injury, and it reactivated a HSV1, common virus that's known for periods of latency. Because a fun fact, many astronauts, in fact, I think most astronauts actually have HSV1 reactivation because of space flight. So when they go into space, it reactivates. So this suggests that head injuries could lead to virus reactivation. Then, because of the virus reactivating, it can lead to things maybe like inflammation, and possibly neurodegenerative diseases later in life. So we still really don't know a lot of why certain neurodegenerative diseases are caused, so this might be one pathway. All right, turning gears here to hornworts. So hornworts reveal a spatial model for pyrenoid-based CO2 concentrated mechanisms in land plants. So hornworts, they're primitive plants that you've probably heard about in intro biology and then never since then. But they have a special property. They have a pyrenoid-based CO2 concentrated mechanism, or PCCMs, and this turbocharges photosynthesis. It's found in no other land plants. So researchers do find this in green algae, and so we've been using that as a model system for these PCMMs. But rather than using green algae, researchers could be using hornwort, apparently, as a model organism, especially to study how to transfer these PCMMs into other plants like crops to improve growth, efficiency, yield, etc. So hornworts have their time in the sun here. All right, on the plant kick here, we have diverse plant RNAs coat Arabidopsis leaves and are distinct from apoplasmic RNAs. Arabidopsis, a model plant. So it looks like there's a lot of RNAs on plant leaves that researchers have found. They come from the plants themselves. They're not just extruded, but they're actually secreted directly. And apparently this RNA may play a direct role in shaping the leaf microbiome. All right, final article for today here. I told you there's a lot of great ones. We're going to wrap up with 2 decades of bacterial ecology and evolution in a freshwater lake. So researchers used 2 decades of 471 metagenome time series data from Lake Mendota, that's in Wisconsin. They found in these bacteria communities, researchers found genetic shifts in the genomes across season and to a lesser extent decade. So like 80% in season, 20% in decade. So essentially, it's as if the bacterial communities were like evolving rapidly for a while and then sort of reverting back to an earlier state, that like their evolutionary shifts across season, I guess in response to ecological changes. All right, let's turn to our research tools. Rick Wicklin from the Do Loop has his top 10 posts from the Do Loop in 2024. There were 4 in SAS programming: understanding the put and input functions, finding the label of a variable, find the name of the variable that contains the largest value in each row, and how SAS displays missing values. There's also 3 from data visualization, 10 tips for creating effective statistical graphics, so a top 10 within a top 10. There's some top 10 inception going on here. Next up, there is use colors to visualize groups in a bar chart and visualize patterns of missing values. Finally, 3 in statistics and data analysis, which I have a special love for. There is what is polychlor correlation. There was the likelihood ratio for linear regression, and finally, estimate a proportion in a confidence interval. 2 new R-bloggers posts that might be of interest to you. First is from David's blog. Your classifier is broken, but it's still useful. So how to use a binary classifier over time properly, especially with the idea of, where was it? That you can't really use precision and recall over time, but you can use specificity to get relatively stable predictive prevalence. All right, the other our blog post from Toma TSQL, little useful are functions, vanishing sentences. So using ggplot2 and gganimate, you can make sentences that the words disappear one by one. All right, we're going to close out our time here with some research funding. 2 new grant opportunities. First, from the NIH, the Trailblazer Award for New and Early Stage Investigators. This is an R21 clinical trial, optional. And it's an opportunity to pursue research programs that integrate engineering and the physical sciences with the life and or biomedical sciences. So a nice interdisciplinary endeavor here. And second, from the NIH, enabling discovery through genomics, and the NIH, or sorry, not the NIH, the NSF. This enabling discovery through genomics. This is called the EDGE program. NSF loves their acronyms much more than the NIH does. Anyways, it's to support research to advance understanding of comparative and functional genomics. Heterobranchia. Thanks for joining me for Science Research Weekly. Science on.